Hi, good evening. I'm Bobby, back with you. Alright, continuing from where we left yesterday. Significance of mitosis, essential for growth and development of a multicellular organism, helps to maintain the stability of chromosome number and hence provides genetic stability. Provides genetic stability. Methods, of multiplication of unicellular organisms. Mechanism for repair of old and worn-out cells. Restores the surface volume ratio of cells. Restores the nucleoplasmic index. Produces multicellular conditions and provides opportunity for differentiation. Repair of body takes place because of addition of cells by mitosis. Types of mitosis. When keratinosis is not followed by cytokinesis, the division is called free nuclear division. Repeated free nuclear divisions result in the formation of a multinucleate cell or a xenocytic cell in unicellular organisms such as amoeba and yeast. Mitotic events usually occur within the nuclear envelope, which remains intact, and the nucleus divides by furrowing along the cytoplasm. Such mitosis is called intranuclear. In multicellular organisms, the nuclear envelope breaks down and the mitosis is termed extranuclear. Replication of chromosomes without keratinosis is called endomitosis or endoploidy. Endomitosis results in polyploidy when chromatids separate from chromosome. Meiosis forms the basis of sexual reproduction and can only occur in eukaryotes. In meiosis, the diploid cell's genome, which is composed of ordered structures of called DNA called chromosomes, is replicated once and separated twice, producing 4 sets of haploid cells, each containing half of the original cell's chromosomes. These resultant haploid cells will fertilize with other haploid cells of the opposite gender to form a diploid cell again. In meiotic cell division, reduction in chromosome number occurs, which gives rise to 4 haploid cells, each having half the number of parental chromosomes. Meiosis was discovered by Strasburger. Terms meiosis were used by Farmer and Moore. If meiosis does not take place in an organism, the chromosome number will be double in each generation. Meiosis must take place in the organism to maintain the chromosome number constant. The cell which thus undergoes meiosis is called a meiocyte. The meiocyte is comparatively large and possesses a large nucleus. It contains a diploid number of chromosomes which form a network. In animals, the meiocytes are the primary spermatocytes and primary oocytes present in the gonads, while in plants these are represented by sporocytes found in the sporogonia. There are 3 types of meiosis: the zygotic or initial meiosis, which occurs immediately after fertilization in certain protozoan groups, fungi, and algae. And now continuing, chiasmata are found in the meiosis of almost all eukaryotic organisms. However, achiasmatic meiosis or meiosis without chiasma has been reported in some organisms, example males of higher diptera, including Drosophila, the Norpus scorpion fly, mantids, mainly mantids, and roaches, some grasshoppers and scorpions. A chiasma formed at the ends of chromosomes is called terminal chiasma. Chiasmata formed along the lengths of chromosomes are called interstitial chiasma. The homologous chromosomes start separating, called disjunction, as the nucleoprotein complex of synapse chromosomes dissolves. This occurs in diplotene. Diplotene is the longest and most active subphase of Prophase I of meiosis. Dicytotene is an arrested diplotene noticed during oogenesis, oocyte stage in the fetal development. In Diakinesis, terminalization of chiasmata occurs. Terminalization is the disappearance of chiasmata of sliding towards the tips of the chromosomes due to tight condensation. The degree of terminalization is expressed by the terminalization coefficient T. The nucleolus degenerates and nuclear envelope disintegrates in diakinesis. Metaphase I. In metaphase I, spindle apparatus appear and their chromosomes are arranged on equatorial plate with centromeres towards the poles. Spindle is formed. Spindle fibers become attached to the centromeres of the two homologous chromosomes. Next is the anaphase I. In anaphase I, the homologous chromosomes break apart, and this process is called disjunction. The separated chromosomes are termed as dyads. At the end of anaphase I, two groups of chromosomes are produced. At two poles have half the number of parental chromosomes. Because the chromosomes undergo crossing over during prophase, the composition of the separating homologous chromosomes is different from that of the chromosomes undergoing asynapsis. In homologous chromosomes, one chromatid is unchanged while the other has undergone mixing of maternal and paternal sections. Right? And the division of centromeres, which is characteristic of the anaphase and mitosis, does not take place in meiosis. Non-disjunction and breakage of chromosomes may occur during anaphase I due to non-dissolution of chiasmata. It produces chromosomal aberrations, aneuploidy and polyploidy. Telophase I. In telophase I, polar groups of chromosomes arrange themselves into haploid nuclei, nucleolus, nuclear membrane, appear at each pole, thus two daughter cell nuclei are produced. Interkinesis is the stage between telophase I and prophase II, where chromosomes are elongated. Centrosomes undergo replication, but DNA synthesis does not occur. Proteins and RNA synthesis occur. It is important for bringing true haploidy. And on MEIOSIS II, is essential to separate the chromatids of univalent chromosomes, which differ from each other in their linkage groups due to crossing over. It differs from mitosis in that DNA does not duplicate, but centromeres do. Meiosis is subdivided into four phases: prophase II, metaphase II, anaphase II, and telophase II. In prophase II, nucleolus, nucleoli, and nuclear envelope degenerates and the chromosomes shorten a little. Centrioles move to the polar regions and are arranged by spindle fibers. In metaphase II, the chromosomes arrange themselves in the center with the help of spindle fibers. The center of each chromosome divides and chromatids separate to form daughter chromosomes in anaphase II. At the end of anaphase II, four groups of haploid chromosomes are produced. The process ends with telophase II, which is similar to telophase I, marked by uncoiling, lengthening, and disappearance of the chromosomes occur as the disappearance of the microtubules. Nuclear envelopes form. Cleavage or cell wall formation eventually produces a total of four daughter cells, each with a haploid set of chromosomes. Meiosis is now complete. And the chiasmata found in the meiosis of almost all eukaryotic cells, eukaryotic organisms. However, a chiasmata meiosis Sporogenetic or intermediate meiosis, which occurs at the time of spore formation, is higher in plants, and gametic or terminal meiosis, which occurs at the time of gapnogenesis, is in a fewer lower in plants, most animals, and many protozoans. Meiosis occurs only in reproductive or germinal cells. Meiosis consists of two divisions, meiosis I and meiosis II. First nuclear division, meiosis is called meiosis I, or reductional division, or heterotypic divisions. Second nuclear division of meiosis is called meiosis II, or equational division, or homotypic division. Meiosis II is of shorter duration than the typical mitotic division. Meiosis I consists of four stages: prophase I, anaphase I, metaphase I, anaphase I, telophase I. Prophase I is lengthy phase compared to mitotic prophase. Prophase I is divided into five subphases. They are leptotene or leptonema, Zygotene or zygonema, pachytene or pachynema, Diplotene or diplonema, and Diakinesis. A large nucleus is seen in the meiocyte during leptotene. In leptotene, the nuclear membrane enlarges and chromosomes become distinct. Chromosomes appear as long and slender threads, single threads, bearing bead-like structures called chromomeres during leptotene. In many animals, during leptotene stage, all chromosomes in a cell converge at a common point near centromeres, centrioles, through a structure called attachment plate. It gives a basket-like appearance. This is called bouquet stage synesis, right? The cells undergoing meiosis have diploid number of chromosomes and are called homologous chromosomes. During zygotene, the chromosomes become shorter and thicker lengthwise. Pairing of homologous chromosomes begins. Each chromosome of the homologous pair is contributed by the parent. Pairing is exact and takes place at the chromomere for chromomeres. Pairing of homologous chromosomes, maternal and paternal, is called synapsis or syndesis and occur in the zygotene. The process of attachment of homologous chromosomes called synapsis leads to the formation of bivalence. Synaptonemal complex is formed of the region of synapsis. This complex has one central and two lateral longitudinal elements which are connected by a lateral element. Each lateral element occurs in between two chromatids of a chromosome, and a central element lies between two homologous chromosomes. Each chromosome of a homologous pair have chromatid known as sister chromatids. Each bivalent pair consists of